Today we will be dealing with the most used machine, that is the Illumina. We will see the video later. So this video link is there. Illumina, we are going to study how the Illumina sequencing technology is done, that we will be studying. So in Illumina, the sequencing reads, the reads are of size of the reads are about 150 base pair. The read length is 150 base pair. Just concentrate on the class. As I complete this Illumina, you will understand. So about 150 base pair reads are sequenced by using Illumina. And what it is the Illumina sequencing is also same as that of the 454 sequencing, the initial steps. What we do is that we will if you have any doubt, you can ask in between. We will take the genomic DNA and we will break down into small fragments using sonication, nebulization, etc. If you can recall last class, you will get it. And next, for library preparation, next process is the addition of adaptase into both sides of the fragments. After addition, what we have done in the 454 sequencing is that it were attached to beads, and beads were to the beads we added oil to form the emulsion. Inside the emulsion droplets, the PCR was conducted. But actually in Illumina, both the sequencing and both the library preparation and sequencing, both will happen in a small glass slide called Illumina flow cell. There is no emulsion PCR, separate emulsion PCR in a separate tube happening in the Illumina. Everything, the library preparation, that is the amplification of the fragments as well as the sequencing, both occur in this flow cell. So we have a genomic sequence, and we broken it down, and we attach adaptase to the both ends of the fragments. It is same for both sequencing technologies. So PCR is needed to be carried out for amplifying each read. So for each fragments, in order to amplify more than 1000 times, each fragment is being amplified, so that it will create a spot, that amplified region, it will create a spot in the amplified region. We can see how it happen. So the amplification of the each fragment need to be done. That process is the library preparation process. So each of these fragments, okay, there we have attached the fragments to a bead, but all these processes occur in a slide called Illumina flow cell in Illumina, where the slide contain a lot of it is attached to a lot of primers, okay, which are complementary to the adaptase. In 454 sequencing, what we have seen, all primers were attached to the beads. A lot of primers were attached to the beads, but here the primers were attached to the flow cell, and the fragments were added to the flow cell, so that the fragments will be base pairing with the primers. And after amplification of the amplification of the one sequence, here in Illumina, bridge amplification occur. Bridge amplification. What it is that actually in 454, what we see after the amplification of fragment, one fragment will separate from here, and they will pair with the next primer and again start, or one fragment will separate from here and bind to the next primer, and the amplification occur. But here what happen is that after the amplification, this fragment, I am just rubbing that, erasing this fragment. This fragment, what it will do? It will form a bridge. It will form a bridge and bind to the bind to the nearby primer. And using that primer, it will amplify. The bridge will be amplified. Using that, I will show it so that you can understand. So here, using this here our sequence attached, and using this primer, our the amplification occurred. Then this sequence, this DNA get this like form as a bridge and a base pair with the nearby primer, and using this fragment as a template, this primer has been extended. This is called bridge amplification. I will show in the video, so you will get understand very easily. So like that, the amplification occur. The amplified fragment forms a bridge and binds to the nearby primer, and further the amplification will continue. So that is called bridge amplification. Bridge amplification is called is happening in the this illumina. So cluster generation or library preparation process we are seeing. The sequencing templates are immobilized on proprietary flow cell surface designed to present the DNA in a manner that facilitates the access of enzyme while ensuring high stability of the surface bound template and low non-specific binding to the fluorescently labeled nucleotide. So what is illumina flow cell? What is this slide? It is not just a small slide. It is just a size of a slide, but where the sequencing chemistry occur. The illumina flow cell is where the sequencing chemistry occur. The flow cell is a glass slide, okay, just a glass slide, which contains small fluidic channels. It contains fluidic channels through which polymerase, DNTPs, buffers can be pumped. This glass slide contains fluidic channels through which we can pump the polymerase, DNTPs, buffers, so that the PCR reaction can occur. The glass inside the channels is decorated with the short oligonucleotide complementary to the adapter sequence. So as I said, the glass slide, attached to the glass slide is a lot of short oligonucleotide, which is complementary to the adapter sequence. So that as I shown here, every the full glass slide is attached by the oligonucleotide, which is or primers, which is complementary to the complementary to the adapter sequence, so that the fragments with adaptase can bind and conduct the amplification process. So this is the illumina flow cell, where the whole process of library preparation, cluster generation, as well as the sequencing occurs. Here the sequencing is not occurred by pyrosequencing. Here the sequencing is occurred by sequencing by synthesis, which is somewhat similar to that of the Sanger sequencing method. So we have just studied about the flow cell, where the sequencing as well as the cluster generation occur. It's just a glass slide containing fluidic channel through which DNA polymerase, DNTPs, buffers can be passed through, and it is the slide is fully decorated with a short oligonucleotide or primers, which is complementary to the adapter sequence. So the process in which process of the solid phase amplification create 1000 identical copies of each single template molecule in close proximity. So in a diameter of 1 micron or less. So the first process is fragmentation of the DNA, and then adaptase are linked. What we seen in that for the 454 sequencing, the same thing we can see here. Adaptase are linked to the DNA sequence. Let me look who all are there. So adaptase are linked. So I will be taking up until sequence analysis, what is the basic things. So please be there if you know this, because many people know how Illumina sequencing is done, everything. So don't go away. So so we fragment the DNA here, and it is adapted attached to adaptase. And we can see the flow cell which contain the oligonucleotide or primers attached. These primers are complementary to this adaptase. So these DNA can attach to this adapter can attach to this primer, and the PCR amplification can occur. So like this happening here in the illumina. So the bridge amplification, as I told, as after one amplification, the one of the one of the the after the amplification, then second round of denaturation occur. After denaturation, the strand forms a bridge and got base paired with the nearest primer, and a bridge amplification occurred. Bridge amplification like this occur. So like this, like the bridge amplification, all this take place in an area of in a diameter of 1 micrometer. Okay, and all this amplification. So lot of bridge amplification occur here and there, here and there. So all these things of single fragment bridge amplification occur in a area of 1 micrometer. So when we see this flow cell from through the detectors, we can see these 1 micrometer region as a spot. So each amplified fragment can be visualized as spots in the flow cell. So each amplified fragments can be observed as spots in the flow cell. So the spot the size is about 1 micrometer. So the all bridge amplification of one fragment occur in a diameter of 1 millimeter. Sorry, 1 micrometer. 1 micrometer. So 1 micrometer or less. So then denaturation occur, then further bridge amplification occur, like this. So in 1 micrometer diameter, one fragment get amplified and form thousands of copies of that fragment. Like this, by bridge amplification, another fragment get amplified and form a spot here, and the spot here. Like this, each fragments are amplified and forms spots throughout the flow cell. So this is how it occurs. This is how it occurs. So we can see after this, after the amplification or cluster generation or library preparation, we can see this process is called cluster generation, library preparation. Then the process of sequencing occur. The process sequencing I will explain soon. So this is how the process occur. So after the amplification, amplification, each amplified region is observed as a spot in the flow cell. So each spot represents a amplified product, thousands of amplified product from a single DNA piece. So there will be a lot of DNA piece. Each spot represents the amplified products from a single DNA piece. So that is how the illumina occurs. So sequencing by synthesis. How the sequencing occur, I will explain now. So this is all about the cluster generation. We have a slide. It is called the flow cell, in which and we first taken the genomic DNA and randomly fragmentized using sonication, nebulization, etc. These fragments are then attached to this flow cell. How they attach? Because the flow cell is being attached to the flow cell. There are lot of oligonucleotide attached here. These oligonucleotides sequence is complementary to the sequence of the adaptase attached to the fragments. So they will base pair, and they will the particular fragments will be amplified, and this amplification occur by bridge amplification. Each denatured fragment will form a bridge to the neighboring oligonucleotide, and then bridge amplification occurs here. So this is how the cluster generation occur in the case of illumina, and each of these amplified products, about thousands of the single fragment is amplified thousands of the times, and each of these clusters will be seen as a spot in the flow cell. So then the sequencing reaction occurs. Sequencing reaction occur by sequencing by synthesis. We have seen in the case of 454 sequencing, the sequencing occur by pyro sequencing. Sequencing occur by pyro sequencing. What the method was pyro sequencing. It measure the pyrophosphate released, pyro sequencing. So there we have just added one nucleotide at a time for the sequencing in the sequencing reaction. If that nucleotide is attached, the pyrophosphate is released, and it will react with the luciferase enzyme and give a color. Here we don't need to think about adding one nucleotide at a time. Here we can add all the 4 nucleotides at a time, and the 4 of the nucleotide will be fluorescently labeled with 4 different colors. We have added the 4 nucleotide A, T, G, C with 4 different fluorescent color. So that is the we sequence in illumina by sequencing by synthesis. We add all the 4 fluorescently labeled nucleotides to the flow cell to sequence tens of millions of clusters. All clusters are seen in the flow cell, and we will add 4 fluorescently labeled nucleotide for the sequencing of the DNA sequence. Okay, during the sequencing cycle, what happened? First, the denaturation of the fragments occur during sequencing in the flow cell. First, the denaturation of the fragment occur, and then the primers will be added, and primers will be added, and then single labeled dideoxy nucleotide phosphate is added to the nucleic acid chain. The nucleotide label serve as a terminator for the polymerization, so that each DNTP incorporation, the fluorescent dye is imaged to identify the base, and then enzymatically cleaved to allow the incorporation of the next base. Since all 4 reversible terminator bound DNTPs are present as single separate molecule, natural competition minimize incorporation bias. So what it is happening is that we have A, T, G, C. Our sequence is A, G, C, T, A. Okay. So C, T, A. So we have all the fluorescently labeled, means A will be labeled red in color, T will be labeled blue in color, G will be labeled yellow in color, and C will be labeled green in color. Okay. So here it is A. So T should bind here. Okay, all these nucleotide has a terminator sequence, terminator label attached to it. All these have a terminator label attached to it. Okay, sorry, here terminator label. So T will bind, and T has a terminator label. The T color is blue. As T will bind to this cluster, a blue color is produced from this spot. A blue color is produced from this spot. And next is C. Then again, then the C can come and bind here. Okay, because the C will base pair with this G. Okay, but when C is trying to bind, the terminator won't allow it to bind. Terminator won't allow it to bind. So after the first round of addition, so after the first round of addition, okay, after the first round of addition, who has been added here? The T has been added here. So we got a blue fluorescence. So as the T has been added here, next before the next round of amplification, next round of addition or next round of sequencing, all the fluorescence available here has been removed, as well as the terminator sequence attached here is also removed. Then fresh nucleotide is being pumped. So terminator is being removed. When the terminator is removed, only next C can come here and bind. C also have a terminator. So next C cannot come and bind. So C has a fluorescence of green. So after blue fluorescence, from that spot green fluorescence came. So like that. So C has a terminator. This terminator is being next round of sequencing. This terminator is being removed, and then only at next G can come. So like that. So each nucleotide has a terminator which don't allow multiple nucleotide to base pair during each cycle of sequencing. So we can see here all the fluorescently labeled nucleotides are here, and here the first nucleotide is added, T, and it is this T is bound to a terminator, so that no other sequence or no other nucleotide can be incorporated to this cluster. So what happened? I will read that image taken on the slide. Each read a location, there will be a fluorescent signal indicating the base has been added. So T has been, the slide is then prepared for the next cycle. Terminators are removed. So after T is been added, terminators are removed from this T, allowing the next base to be added. After addition of one nucleotide, the terminator should be added. Then only next T can come here and bind. And fluorescent signals which is already come for T is already removed, and preventing the signal with contaminating with the next image. So all fluorescent signal is removed, and the terminator is removed, so that in the next round the next T can come and bind. The process is repeated, adding one nucleotide at a time and imaging in between. Computers used to detect the base in each site on each image used to construct the sequence. So you can see in each spot in the flow cell we are getting different colors. Each color indicates a particular nucleotide incorporated. Here in this spot we are getting this color, and then blue color for G, then C, then T. All this color based on the fluorescence of the fluorescence coming, each spot or each cluster is being sequenced parallelly. That is a massive parallel sequencing. Okay. So this is how illumina happened. So we have a sequence of a lot of fragments, and these fragments sequence usually it comes in a sequence format called from an Illumina machine. The sequence comes in a sequence format called BCL format. This BCL format will be converted to the most used format called FASTQ format. This is the most used format used for the NGS sequence data analysis, that is the FASTQ format. This FASTQ format is the so this BCL format which is produced from the Illumina machine will be first converted to the FASTQ format. That is the first step involving in the sequence analysis. So usually the FASTQ format, what is the FASTQ format? This picture we will come later. So this is a FASTQ format. It's somewhat similar to the FASTA format which we seen in the GenBank, all these things. But a FASTQ format, the difference is that it says about the quality of the sequence. It has a line which says about the quality of the sequence. How good is the quality? We can see that from this picture. Yes, from this picture, if some of the peaks are overlapping, you can see here overlapping peaks. If overlapping peaks come, we are confused about the identity of that sequence. We don't know what actually the sequence is there about. If there is no overlapping, if only one colored peak is coming, you can see distinct peak is coming, we can confirm that that is A or T or that will be there. But if the peaks are overlapping, the sequence quality will be less. So like that, from the Illumina machine, the BCL format or base calling format will be converted to FASTQ format, and this FASTQ format is the format which contain the quality of that particular sequence. The FASTQ format consists of 4 lines here. 4 lines. It consists of 4 lines. So what these 4 lines indicate, that we will discuss now. So each entry of FASTQ has 4 lines. Okay. A sequence identifier with the information about sequencing run and the cluster. So this is a title the line where it tells about the sequencing run, which cluster it belongs, deals with the sequencing machine and which cluster that this sequence belongs, all these things. Exact contents of this line vary based on the BCL to FASTQ conversion software used. So based on the which software we use for the conversion of BCL to FASTQ, this line may vary. This line mainly tells about the technical details which sequencing about the sequencing run or about the machine and about which cluster it is. So like that, purely technical things are listed in this header line. That is the first line where the sequencing information is present. The second line is the sequence itself. The sequence itself, the base call, the sequence ATGC, the sequence itself. Sometimes they denote as N, where we can't confirm a particular nucleotide. The peaks are overlapping, we will just add as N because we are not confirming. The third line, which contain a plus sign, which it indicates that it is sequence ends, sequence line ends. See, not sequence will not end. Sequence line ends, or we can say it is a separator. The plus sign is a separator, which is simply a plus sign will be there in the third line. And fourth line is the base called quality score. Usually it is calculated using a software called Fred quality score. Fred plus 33 encoded using ASCII character, it represents the numerical quality score. Usually these quality score were determined by the software called Fred, and is quality score for each nucleotide. The quality score for each nucleotide. The quality score for T is the A, the quality score for T is A, the quality score for this T is A, quality score for G is A. What is this A, G, all these we will tell. So this is how the from the illumina the data comes out as BCL format, and the BCL format will be converted to the most accepted, most used format. From the FASTQ format only every analysis has been done by using different softwares. This FASTQ format is the most widely used format, just like FASTA format for GenBank. It consists of 4 lines. First line will be the identifier line, which consists about the sequencing, what machinery used, cluster, details about the cluster, details about the run, all these things, technical things. Second line is the original sequence. Third line is a plus sign, which is separator, and fourth line is the quality score, which is indicated as ASCII characters. What is ASCII characters? You can see a different character, less than symbol here. So what is this denotes? You can see. So this score is called Q score. This quality score is called Q score. Okay. What is this quality score? The quality score is given score of a given base. The quality score of a given base is called Q score. Each base has a score. Okay. Each base has a score. Here the base T score is A. Base T score is A. Base T score is just a number like it. Don't think it is adenine or anything. The quality score of T is E. Like that. These are ASCII characters. So this is calculated by equation. This is Q is equal to minus log 10E. This E is estimated probability of base call being wrong. So highest quality score indicates smaller probability of error. If the quality score is high, we can say that the quality score is high, which indicates that the nucleotide is there is no wrong in nucleotide in this position. Okay. When we are adding, the PCR is a technique which a lot of error can occur. Okay. So during this addition of nucleotide, error can occur. If the quality score is high, the error chances of error is less. If the quality score is low, the chances of error in calling that base is chances of error in that of that base in that particular position is high. Okay. So high quality score will be good than low quality score. You can see when you are doing Galaxy, for the analysis of this FASTQ sequence, you can see this type of graph where we can see the quality score here. Quality score usually ranges from 0 to 40. Okay. 0 to 40 is the quality score. Usually a good quality score will be, very good quality score will be 30. If the quality score is 30, is very good quality score. You can see the initial sequences here. That is, these are the sequences. Sequence position. Okay, this is the first position. The initial sequences were of a very good quality score. It has a quality score of 36. It has a quality score of 32. It's very good quality score. But when it comes to the last sequence, the error bar you can see at the lower quality area, about 20. 20 is a low quality, you can see. So low quality you can see. So as coming to what 60, 75 base pair, the quality of the that sequence become deteriorating, as we can see from this graph. So as I said, this is the quality score. We need to have a good quality score is around 30 is good. Quality scores start from 0 to 40 and 30 is very good. We can see here the quality score of the initial sequence. This is a sequence from the first nucleotide to 99 nucleotide, and the first sequences were very good quality score. And when it comes to the last nucleotide, the error bar is, we can see it in this region where 20, the quality score is 20, the quality score will be low, so that the sequence at that region is having less quality. Okay. So high quality score means that sequence at the region, their probability of occurring that or that sequence at that region is very good. So there is no this is very proper sequence has been sequence is present there. But if it is lower quality score means that sequence, or if it is A is added there, okay. Actually we don't have a confidence on A to be there. Maybe it can be T or G or C, because the quality score there is less. So the graph may be overlapping, and the quality of the sequence at that position is not good. They may also lead to the increased false positive variant called. We would add A there, but actually that position, 95th position, may be T. So at that area the quality score is less. You can see here there is a symbol. Usually symbols indicates a lower quality score. Here the quality score is less, so the symbols are indicated here. So I will tell what it is, symbol, everything. So here the quality score is less. So here the quality score will be lower, so that we don't know it is C. It can be T. We don't know. The peak here is overlapping, and we can't determine. So it is usually C, the fluorescence of C, but actually we don't have confidence in this region. So that is the there the quality score is less. So the sequencing quality reaches Q30, that is the very good quality. Virtually all reads are perfect. So if the Q30, all reads are perfect, there is no errors in the sequence. And why Q30 is considered as a benchmark quality? This is why Q30, if their sequence quality score falls in Q30, the sequence, all your graph fall above this, your sequence is of a very good quality. So you can see the quality score. If it is 10 quality score, 10 Q10, 1 in 10 chances of error can occur. When it is 30, 1 in 1000, very good, about 99% accuracy we can see. If it is Q10, about 90% accuracy only. We have 99% accuracy in the case of 30 Q score. So from here only we got this A, E, all these things. From this table we got. So you can see different ASCII score and symbol. So the quality score 0 means at that region this symbol will come. Where is that the less than symbol? Yes, the quality score is here 27 only, that less than symbol. Okay, here you can see. I will rub that. Here the quality score is only 27. Okay, here 27 only. So like that, we have an ASCII table which shows where is A. A is the quality score of A is of quality score 32, and E is of quality score 36. Very good quality score is been here for most of the sequence. It's a very good FASTQ sequence. All these sequence are being very high quality. So only one sequence, it is about 27. It's also some acceptable. So if it is if your sequence is of 0 quality score, the symbol come that A. If it is 0 quality score, this symbol will come. So different symbols will come in the fourth line. This fourth line, these symbols can come according to the quality of your sequence. These symbols can come based on the Q score. Okay, I hope you understood. So we are just going through the basics of whole genome sequence data analysis, and then we will see the methodology, how the sequencing is done for very shortly. We will see how the sequencing is done for ion torrent and solid sequencing by ligation. I think sequencing by ligation is not being used today. So these two very shortly we will see, and we will wind up. So actually, in the case of sequence data analysis, we are going to do the sequence data analysis using a online tool called Galaxy. Okay, Galaxy is online, and it is available freely online, and you go here and just to take the Galaxy. Galaxy means use Galaxy. That is the, I think I have done it one experiment before my experiment is coming, I think. So usegalaxy.org is the website. You can go usegalaxy.org, and what you need to do is that you should register here. So I have already registered as Dr. Noble. You will have a symbol like a register. You can give your email ID, and you can register here first. Okay, register here. This Galaxy is just like NCBI for data analysis. It contains a lot of tools which needed for different sequence analysis. In the case of RNA-seq, in the case of microsatellite, in the case of lot of viral DNA, in the case of small exome sequence DNA data analysis. So actually, every sequence analysis follows somewhat a basic workflow or pipeline in the analysis. So we have this illumina machine the FASTQ sequence. From the illumina machine we have the FASTQ sequence. Then what we do is that we will use a series of tools from this Galaxy to analyze our FASTQ sequence. What we need to do, we have only the FASTQ sequence of a number of pieces. We have the FASTQ sequence of the fragments which we have made during the sample preparation, fragments made through sonication, nebulization, etc. Now we need to assemble these fragments. For assembling these fragments, we use the help of a reference genome. If we are doing a human genome sequencing, we take a reference genome which is already sequenced from NCBI or genome sequence databases such as UCSC genome browser or somewhere, and using this reference genome we will find out where which piece belongs to. Okay, where which piece belongs to, and then we will finally find out the whole sequence in the proper sequential order. Hope you understood. So how we will do this? We usually use a reference genome. Based on a reference genome, we have the sequence of millions of fragments which we have produced from the genome. These fragments, where it lies, where it goes, we don't know. So for that we will take a reference genome. We will take a already sequenced genome, and using a reference genome, is a reference genome of human which is already sequenced. Human genome sequence is available since 2003, I think. So we have a number of assemblies available in UCSC genome browser. I think last full genome sequence assembly is from 2013. I think we have seen when we are taking that gene sequence for primary designing, 2013. Newer assembly is there. I think it is not fully one. This is the 2013 one we are using. So using the reference human genome, we will assemble all our fragments, and find out after this fragment which fragment will come, after this fragment which fragment will come, after this fragment which fragment will come. So based on the reference genome, we will assemble all our fragments and make it, stitch it together to form the whole genome. Okay. This is how we do with our sequence. If we don't have a sequence difference, we will go for overlapping sequence and find out, we will find out the sequence. That is called de novo assembly. De novo assembly. Okay. So usually de novo assembly, I am not referring that now. So how, using a reference genome, how we will find out the whole genome, that is what I am going to explain to you. So actually, when we get the FASTQ sequence, we have the quality score of each nucleotide. Okay. Then also we will go for the quality score of the whole sequence. So that process is done by a tool called FASTQ. I think you know that a lot of tools are there in NCBI. BLAST, BLAST. Cluster level is not there, and which other tool is present? BLAST, different BLAST, N BLAST, P BLAST, and primer BLAST, and then OR finder, and then many tools are present. Just like that tools, the Galaxy, that server also have several tools. It is not just I have referred 4 or 5 tools, more than lot of tools needed, specially needed for each type of DNA sequence. If it is RNA, there is different tools are there. If it is viral DNA, it is different tools are there. So I am just telling about the common whole genome or DNA sequence what we are using. So the FASTQ sequence is there. This FASTQ sequence, its overall quality can be assessed by a tool called FastQC. And then if any region overall quality we just find out, and if any region the quality is so bad, that region we can trim and get the good area. So that for that we will use a tool called Trimomatic, which is present in the Galaxy. All this actually Galaxy is a we are very used to the we are very used to the graphical user interface. Okay. So we can't shift it to coding. That is, the people who are very difficult to shift it to coding use this Galaxy. Otherwise, the people who know how to code, they use the softwares which support coding. Actually, the advantage of coding is that actually I will show you, I have just to show what we are doing here using a very small FASTQ file. But actually the FASTQ files are of very big GB size. So we can't it will take, the Galaxy will take a lot of time for each analysis. We need to take all these tools and analyze our sequence. For that, Galaxy take a lot of time. Days it will take, and a very good system needed. But in the case of command based tools, it is more faster compared to that of the Galaxy. And there also you need a system with a very good memory. So what we are doing here, we will just take a sample sequence. We will just take a sample sequence. You will get a sample this sequence sample FASTQ files you can get from a site called Senado. You can see Senado sample file. You can just search sample FASTQ files. You will get from Senado. I think you can see small files which can be which is easy for analysis and studying. You can get it from here. It's MB files only. If you go Geo website in NCBI, you will get the files, but all are very big GB. We can't process properly. So I have just taken a small file in order to show just what it is happening here. Just like a BLAST only, we will upload it and analyze it. That's all. So So, that is what we are going to do. So, what we need to do is that we after entering the galaxy, we need to register as the user. Okay. Then we need to get the data. For getting the data, we can extract data from the NCBI websites using the particular code, the accession number. You can extract the FASTQ files from the NCBI, EBI websites directly. All these are bigger files. I am not going to take that files. NCBI NRES API. I have not said about NRES. NRES is the search engine used in NCBI, just like Google. UCSC browser, you can just export from there. So, I am going to upload from my computer. I have just downloaded from this Senado. I am going to upload a small file from the computer. Upload file. I am going to upload that. I need to choose local files. I think it is in my downloads. I think it is the FASTQ file. I am just uploading. The status it is showing 0%. Let it upload. Let it upload, and then I will come. If you don't register, the uploading will not be happening. So, the FASTQC file FASTQ file we go for quality check called FASTQC. Then if any portions were not showing the good quality, is being trimmed and removed by Trim-O-Matic tool. And after the trim, you can go for one more FASTQC. FASTQC is a tool which tell about the quality control. You can do it in each step, so that we can see the quality, how the quality increased after Trim-O-Matic. You can go for one more FASTQC. One more time you can do the FASTQC. After the trimming the sequence, you can do one more FASTQC. Then we will go for the genome assembly using a reference genome. Usually in the Galaxy we use the tool called Spades. Spades are the tool which is used for genome assembly, and genome will be assembled as a reference genome. And after the assembly, and we got the whole sequence, genome sequence, then we can go for finding out mutations in this genome. This is called variant calling. If any SNPs are there, SNP is another tool. If any SNPs or any variations present, different variant calling tools are present in Galaxy. We can find out different mutations present in our genome. Okay, and then after finding out mutation, we can visualize the our genomic sequence using another tool present in Galaxy called JBrowse. So, lot of tools are there. The basic steps follow the same in most of the whole genome sequencing. In when it comes to RNA-seq, we will do this FASTQC, Trim-O-Matic, everything, but we will go for annotation of that genes, what genes are present. Some extra tools we will use. That's all difference. Extra tools, you can see the pipelines. A lot of pipelines are available in online. So you can see the pipelines which are commonly used for RNA-seq. So just what they do is that they use different annotations. They will find out the functions of the particular gene, all these things by using different extra tools. So actually, what is the whole genome sequence data analysis is that we will check the quality of the sequence, and the main thing is that we will assemble the sequence using either reference genome, here the Spades is using reference genome, or by de novo by using overlapping sequence. So in both ways we assemble the sequence. This is the most important thing in the DNA sequence analysis. And then we will find out variant, and variant calling we will do, and finally the data visualization we will do. And different tools can be used. CSC genome viewer is there. Different tools can be used for the genome data visualization. In Galaxy we use JBrowse. So that is all about the genome sequencing and data analysis. These are the common steps we use in data analysis. This we will follow in every software. So that's all. So I think this is still 0%. Done. No, start. Oh, sorry, I have need to give start. Sorry. So I have done, and you can see my then close. Here these regions we can see the tools, and here these regions you can see the history what I have done, and here these regions you can see the results which we get. So we have uploaded that sequence here, and then we will just go through the FastQC just to show what is happening here. You can just type what tool, spades or what tool you require. You just type the tool, just upload that thing in the tool, that's all. So FastQC, this is a tool, just select it. So our sequence is already uploaded there. Make other things as adapter list, contamination list all as default, and just do the FastQC, and you will see the quality of the that FastQ file which we have uploaded. So that is what we are doing in the Galaxy, and this is what our whole genome workflow is about. This is what we are doing in a very old genome sequencing data analysis. People do with different tools, and people do with mostly with the command line based tools, so that it will be more faster compared to that of the Galaxy tool. You don't need to worry, you can use Galaxy tool. It will take a bit time only. You can use Galaxy, and you can just get your FastQC, and it is already added to the queue, and it will take bit time. We can continue with our next sequence.